Some guests need no introduction, and this is one of them.
My guest today is Dr. Tony Atala.
Dr. Anthony Atala.
Dr. Anthony Atala.
Dr. Anthony Atala.
Dr. Atala presides over the world's largest lab devoted to bioengineering body parts.
He's the man who's been growing human organs in the lab while the rest of us are still trying to process the idea. A surgeon, scientist, inventor, and world-renowned pioneer in regenerative medicine, Dr. Anthony Atala is the kind of guest you bring in when you want to talk about what's next, because he's probably already building it. As the founding director of the Wake Forest Institute for Regenerative Medicine, Dr. Atala leads the team responsible for some of the most jaw-dropping advances in the field. 3D printing of human tissues, lab-grown organs, and cell-based therapies that are reshaping what's possible in medicine. His work has appeared in over 800 publications, 250 patents, and Time Magazine's list of top medical breakthroughs not once, but twice. In this episode, we talk about more than just technology. We talk about vision, what it takes to lead a global movement in bioengineering, how to turn science fiction into science fact, and why the future of medicine isn't just coming, it's already here. Please welcome the innovator, the icon, Dr. Anthony Atala.
In the not-too-distant future, a revolution is brewing, one that will change the very fabric of our existence. It's a tale of cells that hold the key to unlocking the mysteries of life itself. Let us venture into the unknown, for in this age of stem cells, anything is possible.
I've seen some beautiful...
But we are sitting here with greatness, folks.
Oh, you're
absolute greatness.
That's absolutely not true.
So I witnessed you getting an award yesterday, and you get up there and humbly say you don't know why you're up there. But I don't know how somebody who has done all of the things that you've done. Taylor and I were just going off your bio and just kind of reading all of the amazing things that you've done in your career. So I need to start somewhere at the beginning. You are a trained urologist.
That's right.
And you're seeing patients every day. So how the heck do you encounter stem cells?
Yeah, it's interesting, right? So, I went through my usual training in medical school.
Mm-hmm.
I then, went through surgical internship, specialized in urology, as you mentioned, urologic surgery. And then I specialized in pediatric urologic surgery at Children's Hospital Boston. And when I went through that process, my boss-to-be, Dr. Alan Reddick, was a surgeon-in-chief at Children's Hospital and Harvard Medical School. He basically called me up and said, we're actually going to add research experience to the training. And because we're doing this late in the game, I'm going to give you a choice. You're either you can take just the clinical track or the clinical track and the research time, the research year. And I said, Dr. Reddick, I'm just interested in clinical care. I do not want to do the research year, so I'm going to take just the clinical track. And he said, really? I said, yeah, really. He said, well, I really want you to think about it because I think you may enjoy the research track. And so why don't you think about it, and I'll call you back in a week. And I said, okay. So I talked to my professors, my mentors, my wife, my colleagues. A week later, he called me at the set time. And, I had really given it a lot of thought by then. And so he said, well, what will it be? He called me up and said, what will it be? Are you going to do the clinical track or the research and clinical track? I said, Dr. Reddick, I've really given it a lot of thought, and you have such a great thought about that, but I still only want to do the clinical track. And he said, really? I said, yeah. He told this story all the time. And he said, that's fine. He said, is your wife home? I said, yeah. So he got on the phone with my wife, Catherine, who's actually a psychiatrist, and she said, she got off the phone and said, you really ought to do that. And I said, so he talked to her and convinced her that that's something I should do. So he gets off the phone. She looks at me and says, you really ought to do this. So I said, why? He says, because he thinks that it's going to be worthwhile. So I did show up at the lab there, but it was just fairly much an empty lab. People were just starting out. The technician had been hired, but the faculty member had not yet arrived. And so I had this unusual opportunity to pick what I wanted to do. Mm-hmm. And so that's how I ended up doing research, and specifically tissue engineering and stem cell research.
So I guess the lab was already set up to do tissue engineering? Is that?
No, actually, no. The lab was an empty lab.
Okay.
Entirely empty. A technician had been hired. The faculty member had been hired, but he was working on prostate cancer.
Okay.
And he was not coming in until late August. This was early July. And he had hired a technician to come in and start filling up the lab. And the lab had been doing some work in the area. There was a lab experience there that had to do with fetal surgery.
Okay.
But this was really, kind of saying, well, what project am I going to do? I want to do something interesting. And so my first project really was to, start thinking about creating a new bladder, because, patients in urology who need bladder reconstruction, they typically use a piece of intestine that shouldn't be there. And you start absorbing things that should be excreted. So I wrote in my lab notebook, can we create a bladder using, the patient's own cells? And so the faculty member did arrive 6 weeks later, and he said, well, do you want to help me in my project? And I said, actually, I already have one. Would you help me with mine? So that's how, and then, luck would have it, we ended up implanting that engineered organ 12 years later.
And that was in 1999.
Yeah, actually, yeah. So, finished experience 12 years later. We did it in 1999. That is correct. So it was 9 years later after we started the project.
I interviewed Dr. Charles Cox yesterday, and I asked him, what advice would you give someone, young person, looking at what they're going to do in their career? And he said, you have to just pick a big problem. So you picked, I got to make a bladder. Out of what? How did you do it?
It's interesting. It was very difficult. In fact, I always say, as you know, there are, this is early 1990, 1990. Yeah. So at that time, the term RegenMed had not yet been coined. Stem cells had not been discovered. Cloning had not been discovered. So it was really very kind of science fiction-like type of stuff. And but it was very tough because you couldn't get the cells to grow. I mean, most human cell types back then could not be grown and expanded. Yep. That was just a fact. In fact, the only cells that you could grow and expand outside the body were skin cells. Mm-hmm. And Boston had a history with that because, they were trying to grow skin cells for quite some time for burn patients, not for skin replacement, but for a temporary graft, And just at the heels of when I started to work, cartilage cells could be expanded as well. But pretty much those were the only major cell types that could be expanded outside the body. So we went through a lot of time and effort trying to figure out how to get these cells to grow and expand, and basically, doing growth factor biology at the time. And then, of course, now if you fast forward, 35 years later, most human cell types can now be grown and expanded outside the body.
Isn't that amazing? Yeah. So what was was there a light bulb moment for you when it comes to, everyone in the field puts your name together with adult stem cells. I mean, you are one of the core pioneers in this field in terms of translational research, like research that actually means something to a patient or has an impact to a patient, which we'll talk about kind of has really has been the foundation to your entire career and what you're pushing other people to do too, to think about making things that are going to be usable, not just making things to make things. So when was your aha moment? Because I don't know about you, but when I talk to people about stem cells, I educate a lot of people and a lot of doctors most of the time, and I can see it when they get it. when they finally get it? Like, when was that moment for you?
Really, because I'm trained as a surgeon,
Yeah.
I think the one thing that's motivated me my whole life has been making patients better. I mean, that's really where it came from, right? It came from clinical needs. What is the clinical need for the patient? And then work backwards to try to solve it. And that's really what inspired me, and it still inspires me today. And that's why I still maintain my practice, even though I still do research, because I think that one-to-one interaction with patients and their families and being abreast of what's happening in the medical field really gives you a window into what the needs are. And for me, those aha moments are basically, there's a need here. That's an aha moment, because you say there's a need here that needs to be fulfilled, that needs to be filled. The current standard is just not the best. How can you make it better?
So the first cells you ever worked with were derived from the amniotic membrane or placenta?
No, actually the strategy was that we went to the same tissues, so the same patient. So we would take a biopsy from that organ. But you're right, we did work with amniotic cells, and we can talk about that as well. But the initial work that we did was you are trying to target a patient's organ. And so you go and take a small biopsy of that organ, very small, less than half the size of a postage stamp.
Oh, wow.
And then you're able to get those cells from that organ and expand those organ-specific cells outside the body into large quantities.
And does it sit on a scaffold?
Well, not initially, but
Okay.
you're right.
Yeah.
Once we expand the cells in large quantity, the key is to make sure you can expand those cells. And that was a challenge back then.
Mm-hmm.
You couldn't get these cells to expand, so you really had to go back to biology 101, growth factor biology, and what does the embryo do to develop cells, and how can we replicate that in a cell culture plate. And so once we were able to figure out how to get the cells to grow and expand, then picking the right scaffold or material that will replicate the organ shape and form and the glue that holds the cells together. Because every organ in your body is basically made up of the cells that you have and the glue that holds the cells together. So the scaffolds are the glue that holds the cells together. It's like a piece of your shirt or your blouse that you are able to shape into the organ structure, with one major difference is that all these materials are biocompatible and biodegradable. In other words, they go away over time. They're not going to hurt you inside your body, and they're going to go away over time. So they're made with the very same materials that we use for stitches in surgery. The stitches that you put in then dissolve on their own over time.
Yeah.
So what happens is when you place these cells on these three-dimensional scaffolds in the shape of the organ, it's like baking a cake, if you will, right? You're you're layering these cells on the scaffold one layer at a time, putting it in this oven-like device, letting it cook, if you will, taking it out, and then put it back into the same patient. And because you're using the patient's own cells, there's no rejection.
Right. So was this process, I imagine this process was very long, took very long. Is it the same? Has it sped up?
Yeah, it's a complex process because there are a lot of steps along the way. But once you know the process, then it becomes easy because it's just a protocol that you have to follow. But the whole process, from the time you take the piece of tissue from the patient to the time that you can actually deliver engineered tissue or organ to the patient, it's only about 4 to 6 weeks.
That's incredible. And so you have essentially a bioidentical organ going back in. Is this organ good for life?
It is good for life. Certainly at least for several decades, as we now know from the patients that have been implanted with these structures. We now have 17 applications of our technologies in patients in various tissues and organs. So we've been able to now transfer 17 technologies to patients using these strategies.
Yeah.
But at the end of the day, because the patient's cells are being used, the body recognizes that structure as their own. And the material that we put in, which is the glues that holds the cells together, they're degradable, as I mentioned. They go away just like stitches. But what happens during that process is that the cells that you put in with that scaffold, with that material, once the cells sense that the material is giving way, the cells do what they do in your very own body, which is they make their own bridge.
Yeah.
So 6 months later, you're left with your very own cells and your very own bridge. The material that you put in is totally gone.
Wow. So were there stem cells combined with it, or just the tissue from that organ?
So the nice thing is, they're basically adult cells.
Yeah.
And you are targeting the adult tissue-specific stem cells for that organ.
Okay.
So in other words, they are already, so that bladder cell knows it's a bladder cell. The skin cell knows it's a skin cell. The cartilage cell knows it's a cartilage cell. You don't need to drive the cell to become a different cell type.
Right.
Which is very helpful because you're skipping a lot of steps, and you know that what you're getting is exactly the same type of cell that belongs there.
Yeah. So you have, fast forward a little bit, you have been involved in developing a 3D printer, but it prints organs and tissues?
Well, that's interesting. The development of that was very interesting because certainly the concept of printing had been around for a long time. The concept of printing genes had been around. Printing cells had been around. The question was really how to create structures that would be engineered tissues, really, that would have the structural integrity necessary for the creation of the organ and the implantation of the organ. And that's really what we worked on for many years. And we started out by just using a desktop inkjet printer that we modified with a 3D elevator so that every time the printhead would go through, the 3D elevator would lower itself. And we use, instead of using ink, we use cells with a hydrogel. And, techniques have been around. We just basically, focus those techniques to create tissues that we could implant, viable functional tissues that we could implant inside the body.
That's amazing.
A great team effort, team effort. We had many, many people working on that project.
Wow.
Including Tao Zhu, who was a fellow in our lab.
So you had, in all these years, is this readily available? I'm talking about the bladders, for instance. Is this technology readily available now, or is it there's still a wait?
So it's interesting. We had known then what we know now. We would have never started with a complex organ, level 3 organ. So organs are flat structures like skin are the least complex architecturally because they're flat. They're all complex, but architecturally they're less complex. Tubular structures like blood vessels, urethras are the second level complexity. Hollow non-tubular organs like the bladder, the stomach are level 3 organs, which are more complex. And the most complex organs are the solid organs like the heart, the liver, the lung, the kidney. And we've used different strategies. So we keep doing this in patients, in terms of what we do. And we're doing a lot of these structures. But definitely there's a level of complexity as we engineer these that is, that makes it more complex as you go up that ladder.
So is it a wait until patients will be able to readily access?
So, well, it goes through a clinical trial. So we have things that are in clinically available now. We have things that are in phase 3 clinical trials, technologies in phase 2 clinical trials, phase 1, and of course our pipeline.
But you can definitely see the impact of this work because, to say that the problem you were trying to solve was to build a bladder is not that. It's like we have a national organ shortage, and I see that this technology is going to make a huge impact in our accessibility to compatible organs. It's going to change everything.
Yeah, so, and that is why I think it's important to keep increasing the number of technologies that we create. As I mentioned, we have 17 different technologies now, including skin, cartilage, muscle, urethral organs, vaginal organs, that kind of thing. So we keep increasing the number of technologies that we can offer patients. But the goal for us is to make sure, and for the field really in general, is to make sure that we can tackle more and more of these organs and that we can expand the number of patients who can receive these technologies for their particular indications.
What are you, so you are not only the director of the Wake Forest Center for Regenerative Medicine, but also AFIRM. You founded AFIRM, which in your talk today I learned a lot about. It is mammoth. in all of the groups that you've put together to try and take an innovative idea all the way to the end. End meaning make it available not only to the military and the people that need it most, but also for civilians at the end of the day. What was your inspiration?
Well, so the Armed Forces Institute for Regenerative Medicine was actually, the brainchild of Bob Vandry and General Martinez Lopez at the DOD. And basically, Dr. Vandry had heard me give a talk at one of the meetings, and I kept hearing in this meeting, there's this colonel who wants to talk to you. And, over like a day and a half, I kept saying this, and we kept missing each other. So finally, he connected with me at this meeting, and he said, I saw your talk, and I see that you're doing this stuff for patients, and in terms of engineering tissues and organs, and, we need to do this for our wounded warriors. So General Martinez Lopez had already been scheduled to come to Boston for a visit, to visit Dr. Judah Fogman, who was the founder of the field of angiogenesis. And in that visit, they added me on the list of people to meet with the general. And there were many people that were really behind this. Colonel Bob Vandry, General Martinez Lopez, General Schoonmaker, who actually came in after General Martinez Lopez, who then became the Surgeon General. And then there was this big effort to create this institute. The very first investment on this was to the Regenerative Medicine Foundation, which we created specifically for these funds, where they gave a $5 million grant to establish a first consortium on Regen Med. And then that was a precursor to what we now know as AFIRM, which is the Armed Forces Institute for Regen Med. So it was really, they basically said this is something we need to apply to our military for the future.
And since then, you showcased a few products. So out of all the products that AFIRM has seen come through, I mean, it's just incredible the things that have been innovated through that. What product are you most excited about?
Well, that's a tough question.
Very
non-answerable, because it's like saying, who's your favorite child? But, it's been a great program, really an amazing program, because, it's bringing teams together, and really it's all about teamwork, and it's about making sure that we get solutions for our wounded warriors, and about delivering products to our wounded warriors. And also, like we mentioned today during the talk, is also about making sure that these products get commercialized so they get available to everyone, our wounded warriors primarily, but also to the population at large.
So yesterday, sometime during the talk, someone had mentioned that the arm transplants, and Taylor said, is that true? Can they really do arm transplants? I said, I don't know, that sounds rather complicated. And then today, that came from AFIRM. That was part of an AFIRM project.
Yeah, it was a major AFIRM project in terms of developing techniques to do that and better ways to avoid rejection. It was a program that came out of the first rendition of AFIRM.
Wow.
And it was established in 2000. AFIRM was established in 2008, and there have been several renditions of that program.
Yeah. Well, congratulations. That's an incredible organization. All of the things that you're doing. You've not only founded that, you have a team of 400 people that you manage at Wake Forest. You have published over 800 journal articles, 250 patents, editor of 25 books. How do you have the time to do all this? Do you sleep?
Not much. That's actually a fact. I never needed much sleep because surgeons usually don't. But I do sleep, of course. That's and everything. Basically, AFIRM was founded by a team, right? Mm-hmm. The institute is just a major team, and it's all about teamwork, really. And I've been very lucky and blessed to have an amazing team everywhere, and basically making sure that people can work together to get to where they need to be and to make a difference in patients' lives.
So, Dr. Atala, don't you think it's amazing to look back, because even in my comparatively short experience in the cell therapy world, like almost every major medical school now has these centers for regenerative medicine. That's becoming a thing that we readily see now. Did you, when you first heard the word regenerative medicine and whoever coined that, did you ever imagine that it would get here? Did you think it was a foregone conclusion? Of course we're going to get here. Or were you, what were your thoughts?
So it's interesting, right? So, it's evolved, right? Because really, as you know, there was a field of cell transplantation, right? We can even go further back. We can go back to the early 1900s with Alexis Carrel, who was at the Rockefeller Institute in New York trying to keep organs alive. Charles Lindbergh actually worked with Alexis Carrel trying to keep these live organs, going for, months at a time so it could be transplanted. And of course, then came cell transplantation and then tissue engineering. And the RegenMed, the term RegenMed actually was first used at a medical conference, but not related to what we do today. The term related to what we do today really came from Bill Hasseltine, who was the CEO of an entity that was based in D.C. looking at genomics. And I did get a phone call from him one day. And Bill said, Tony, I'm Bill Hasseltine. I'm the CEO of this company. He used to be a professor at Harvard as well. And he basically said, I'm thinking that we need to start this journal called the Regenerative Medicine Journal, and we need to start a society called the Regenerative Medicine Society. So basically we got together, did just that, started some meetings, started a journal on that, and then that kind of all got merged into what's now called the Tissue Engineering and Regenerative Medicine International Society, which was a separate society on tissue engineering. So the term has really evolved over time, and it's evolved because it's more than just engineering tissues, right? It's using the stem cells that you were inquiring first about, the stem cells, like the amnion stem cells that we work with, or the cells that you're working with, and so many other bone marrow and cord blood cells, et cetera. It includes wound regeneration. It's just a broader term that just captures so much more than our past dictated.
And suddenly you see. Well, let me ask you this question. So when you started to research stem cells, and I guess somewhere along the line you made your first lecture about stem cells, and suddenly you're the stem cell guy, and your peers don't know anything about stem cells. Do they think you're crazy, or do they think you're onto something?
No doubt they thought I was crazy. In fact, no doubt. In fact, I remember the very first time that I submitted a paper for a presentation at our national meeting, and it was about putting these cells into a scaffold and showing that the scaffold was regenerating the cells and creating tissue. And so I submitted that paper, and the paper was actually rejected, and I couldn't believe it was rejected. So I went to the person who was running that meeting, the program chair, because I saw him at another meeting. So I walked up to him and I said, can I ask a question? Because I submitted a paper and I was surprised that it got rejected. And he said, well, which paper was that? And I said, well, the paper we showed that we could grow these cells and put them into scaffolds. And they would, oh yeah, I remember that paper. I said, well, why did it get rejected? Oh yeah, well, because it can't be done. So what do you mean it can't be done? We just show that it could be done. Why are you saying it can't be done?
Yeah.
So the funny thing, that person was actually in Houston,
oh,
believe
really?
it or not, where you are. He was a faculty member in one of the programs there, and about 15 years later, I was a visiting professor there giving a talk, and he was in the audience, and I reminded him of that moment. But it's like anything else, I think all these fields, when you get started, things seem too far out of focus, and obviously there's a very healthy degree of skepticism that needs to be there before the field really progresses.
Are medical schools, do you see medical schools actually teaching about stem cells now, or is that still not in the curriculums?
Oh yeah, stem cells are definitely part of the medical
okay.
school curriculum now.
Wow.
It's really amazing how far we've come.
That is.
with that area, and same thing, with the placental cells that we, the amnion and placental cells that we published on, and we found those cells. And same thing, right? For the first few years, people thought that it was just voodoo, no such a thing as these cells coming from the placenta or the amniotic fluid, which of course now are being used.
Like everywhere for so many applications. But it's still, there are still considerable, very reputable academics that will tell you otherwise. They will say, no, this will never go anywhere. But do you think that we are, do you feel like we're living in the stem cell era? Do you feel like this is, the momentum has brought us here now, the whole field, or do you think we're still far, far behind actually seeing this part of medicine?
I think we're in a very exciting time right now.
Okay.
I really do, because we're at that point where the field is, it's been an emerging field, and we are seeing the field finally come into its own with real valuable, life-changing therapies that are out there right now in the regen med space. Patients who are walking around with engineered organs, and patients who are walking around with better outcomes because of some of the therapies they're receiving. But it's like anything else. What's happened in the field is that you do have this fringe group out there, right, that's offering these unproven stem cells, and they're advertising in the paper these cells can cure everything, and we just have to be careful. And I think that's what's been the problem in the field, is that we do have this other group of folks who are trying to push unproven therapies, and we just have to be careful that we keep making sure that we push the right buttons to make sure these are therapies that are tested correctly, like what you're seeing at this meeting, right?
Right.
And people doing responsible research.
I mean, the sheer amount of like base work and years of toil it takes before that gets to the first patient, no one sees. Nobody sees.
Nobody sees that, right?
Yeah.
It's taking, it takes 20 years
Yeah.
before you can get some of these cells to therapies. It really does, right? It is, and people don't see that. And but they are seeing some of these clinics out there that are charging, outrageous amounts for something like PRP,
Right.
which is a blood product, right? So there are out there people who are trying to make a business out of this thing in terms of direct patient treatments with unproven therapies. And of course, I think that's given the field a little bit of it's affected the field in terms of the consumer not knowing what they're dealing with.
Definitely. It actually tainted the words regenerative medicine. At some point, like you didn't even want to be associated with regenerative medicine because you don't want to be bucketed in with those people. And so the skepticism that's out there, any average physician who doesn't know anything about what we do or research will automatically say, oh, they're stem cells. That's not ready for prime time, because they're bucketing everyone together. And so, it segues to the last time I saw you was in Washington, D.C., and I was so happy to see you there, because I said, they have the right person there. And we got to meet with the Secretary of Health and Human Services. And, after all of that, do you sense that, given this issue with the fraudulent products that are out there that we're inundated with, I have always thought that they're using loopholes in the current regulations, and that the current regulations do need to change. I mean, we brainstorm ideas all the time. We don't know what the right answers are, but what are your thoughts about the current regulations, the regulatory environment? Do you think those are going to change? Do you think they need to change? What are your thoughts?
Well, one of the favorite things that I like to say is that the only constant we have in science is change, right? So we constantly have to change based on the new evidence that we have, based on the new findings that we have. And, regulations sometimes don't keep up with those changes.
Right.
Right? So, and we've seen that time and again, right? It's not just unique to the stem cell field. I mean, you've seen regulatory changes in all areas. And so as the stem cell field has changed and the RegenMed field has changed, regulations also will need to change because they have to address the new findings of the field. And so it is imperative that those changes take place based on the new reality of our field. And so, but that's not unusual. It's happened to other areas as well in the FDA. And the FDA has always tried to protect patients, which is what the FDA needs to do, of course. And we want to make sure that we protect patients, right? But sometimes those changes are slow to come because it takes years for those changes to go into effect. And I think it's a good thing to look at those changes and how can they be accelerated so that we meet our today's reality. And not just for stem cells. As you know, HHS is also interested in doing away with animal trials. Mm-hmm. And so one of the areas that they've expressed a lot of interest in has been the body-in-a-chip work, right, where you're using miniature organs, the size of a pinhead each, miniature kidneys, livers, lungs, brains, to actually do the drug testing on these patient avatars that are using, normal human primary cells. And they have a major interest in that because, as we've also learned, pretty much everything could be cured in a mouse, right?
And we're not animals.
And we're not animals. We're not mice, right? We're not mice. We're not rats. And you saw, it's a major paper in Science maybe 2 decades ago now already, talking about the fallacy of the mouse model, right? And yet we still using the mouse model, and the FDA is still requiring these animal models when they have very little translation to the human equivalent. So HHS is also interested in this area of body-in-a-chip technologies, and so we can actually move that along as well. And they've already made some changes there. As you know, they just came out with this new designation that you don't necessarily need animal studies for some types of technology. So again, that's moving, and I think it's moving in the right direction based on the new evidence that we have with these technologies.
And I also heard that the new FDA commissioner had said something along the lines of, if a product, and not just stem cells, or just any innovative product, I think if it has a certain need, the product, as long as it's shown safety and preliminary efficacy, I mean, we all argue about what is preliminary efficacy, but let's say it meets that threshold, that it can actually be available on the market, but collecting real-world evidence. I mean, our medical records are now all electronic, so we should be able to, the government should be able to collect that data and not have pharmaceutical companies pay, or companies like us pay to run huge trials and collect the data and hand it over. They can actually just get the readouts themselves. They'll understand what safety looks like, they'll understand what efficacy looks like, and then be able to approve products. What are your thoughts on that kind of approach?
Yeah, it's interesting because, as you know, the New Century Act of the FDA that was put out a few years ago, and we were actually, there was a bipartisan group for that. I was fortunate to have been involved with some of those discussions back then, through the Bipartisan Policy Council that actually proposed the new changes that the FDA did, which was the New Century Act of the FDA.
21st Century Cures Act?
Yeah, the 21st Century Cures Act.
And that was like things like RMAT designation and
accelerated
RMAT
approvals
designation, and things like that.
Accelerated approvals, also clinical trial, right, what you just said.
Yeah.
Part of that language.
But that's what it was written for.
Yes, part of that language was actually to do just that. Yeah, and so so I think these are all, again, positive developments, right? Because these are things that have been studied thoroughly. There's already legislation out there that was pointing in that direction. And how do you just keep pushing that forward, but also making sure that you keep patient safety paramount?
Right. Yeah.
Yeah.
Are you active in the legislative world? Do they ask you your opinion all the time?
Well,
Ask you for help?
Well, certainly, I'm sure that I'm not the only one that's being asked. But it hasn't been. It's been an interesting interesting few decades. As the field has evolved, there's definitely been a lot of inquiries from our legislators on the field, and certainly the same continues now. And they do want to hear our feedback, in terms of technologies and what to do with them. But it's one of those things that it takes a collective, the collective us, to be able to speak with one voice and to make sure that we can advance these technologies while preserving patient safety, which of course we all want. We would want that for our loved ones. So it's a fine balance, and I think that legislators are, and the government in general, has had an interest in this. Of course, interest comes and goes, right? There are waves of interest around this topic. But it's good to know that the interest is still there.
Another factor to this, not only the safety and the efficacy of a product, but cost. The way we regulate these products will ultimately determine the cost of these products, right? The longer we are in clinic, the longer we are demanding large-scale trials, it will drive up the cost and at the end of the day, impact the patient. Your thoughts on that?
Absolutely. I mean, one of the number one challenges, as you know, is patient recruitment.
Yes.
It's a major challenge. As much as you think that patients really want these technologies, the fact is that once you start a clinical trial, there are all these exclusion criteria and all these hoops that patients have to go through to enter these clinical trials, and it requires major expense. So there is a cost associated with that. And these clinical trials often last much longer than people think. And of course, there are fixed costs at these companies, right? They're fixed costs. I mean, they're paying their manufacturing facility, their manufacturing team, their clinical trials team. They're paying their entire team. And if a clinical trial is going to, take, 1,200 patients of recruitment, and you're estimating it's going to take it's going to take years to recruit that number of patients. And it's going to take, and it never takes the time you think. It's always longer. It's always you have to contract separately with each hospital and each institution. There's legal paperwork. The costs are astronomical, as you know. Yeah. The estimate, as some of the estimates were that the average time for a product to be approved by the FDA was 18 years at a cost of over $2 billion. Mm-hmm. That's a lot of money and a lot of years. Yeah. So how can we actually, shorten that timeline and shorten the cost so that these technologies are available to patients sooner?
Yeah, while maintaining safety. And I think that's been my argument. It's like, no matter what happens on the government side, we're going to keep going the way we're going. And I just know with certainty that it's going to cost this much if I keep going down this trajectory. And if one were to ask me if I go down this trajectory or perhaps one where we are able to make it available, perhaps we're not allowed to make it available for a profit, maybe we can only make it available at cost or no cost, but we have to collect the data and do this. If we did it this route, I can guarantee that the cost will be significantly less than if we take this route. And if we can achieve the same level of safety, then isn't this worth looking at? Yeah.
Yes, absolutely. I think, this is a good analysis, right? I think in terms of the cost analysis that you're bringing up, what you're bringing up is really a good thing because, having a cost analysis of these technologies is extremely important to make sure that we keep our competitive edge. For example, in Japan, as you know, they're, accelerating all kinds of products. Right. And so one of the, that will mean that industries in the U.S. who are working on similar technologies there will be products out there worldwide before the products from the U.S. get out there. And so the question is, what can we learn from countries like Japan who have taken this much more aggressive approach for approvals? And also, what can they learn from us too, right? Because in terms of preserving patient safety. So I think it's, we're, as an academic, and as I mentioned earlier, I always like to say the only thing that's constant is change. And how can we keep changing for the better, no matter what we do, no matter who we are, whether we are in the regulatory side, in the industry side, in the academic side, in the government side? How can we all keep learning together and have that team approach to keep the end goal the same, which is how can we make patients better?
So then to end it, what would you say to any innovator? Can you list the top 3 things that they should be thinking about to do just that, to get something to a patient, to help a patient? What are the 3 things that they should be there's a zillion things they should be doing, but what are the top 3 things that in your mind, that they should be focused on?
So I think the first thing in innovation is keep an open mind.
Oh, okay.
It's the first thing, right? And, if you want to keep your parachute, make sure your parachute stays open. Make sure your parachute opens up so that you can land safely. So keep an open mind. The second thing is, make sure that you don't overlook the opportunities to take action on those changes that need to be made.
Okay.
And recognizing what those actions are that will try to get you there. And finally, trying to make sure that you explore every avenue to see whether what you're pursuing can actually work and get it there. And I think those are the 3 things that, when I talk to my students or team members, it's very easy to dismiss the negative outcomes of an experiment, right? But the negative outcomes of an experiment are telling you answers. That's how penicillin was discovered. All these discoveries, right? That people, when you look at the history of innovation, it is those unusual findings that led to the innovation, right? Yeah. It was Fleming recognizing that there was these cultures that got contaminated, and he saw that when the area around the contamination, there was no bacterial growth, and there came penicillin. Yeah. Or how Teflon came about, where they put this stuff in this in these reactors, and all of a sudden this liquid sticky stuff came out, and now we have Teflon, which is being used all over the world. Or how the sticky pads came about, right, which was supposed to be a glue substance that didn't work very well. It's amazing, right? All these amazing products that have come out, and these great advances that have come out, because you're looking for the unusual, you're keeping an open mind, you're seeing what the outcome can be, you're seeing the potential outcome, and you try to see how you can keep going to get to your end point.
Well, Doctor, thank you for keeping an open mind in this space. I really believe that you have quite literally led us all here. And so just grateful for you and grateful for this time. It's like I told Taylor, this is for me a once in a lifetime opportunity, I think, so thank you so much.
Thank you so much. Yeah. And thanks for all you're doing. I appreciate it. And thank you for your kind words. It's all about our teams. Thank you so much.
Thanks, Dr.
Appreciate
Teller.
it.